Let's talk about transcription and translation within a prokaryotic cell. To start off, transcription is the synthesis of mRNA complementary to DNA strand. The first step in transcription is initiation. Sigma binds to RNA poly, initiating the binding to the promoter region, tightly causing separation of DNA strands. The closed complex becomes an open complex by tight bonding. Any exposed base pairs are the ones that are going to be copied. RNA poly moves along the DNA strand, and the RNA chain starts to be built one ribonucleotide at a time. Several are added before the RNA poly leaves the promoter region. Promoter clearance is at the end of initiation, and sigma will dissociate. Elongation begins. RNA poly moves along DNA strand, separating the double helix 5' to 3', adding one nucleotide at a time to form an RNA strand in the 3' to 5'. The RNA will be proofread as it moves along, fixing any mistakes made. The two DNA strands anneal behind as the RNA poly passes by. RNA poly will come to a termination region and termination will begin. In prokaryotic, there are two types of termination, row-dependent and row-independent. We chose to describe row-dependent. A ring structure protein called row will come in and bind to mRNA and follow behind RNA poly as nucleotides are added. A hairpin forms, which contains lots of G and C nucleotides that cause strong bonding between each other and form a loop. The hairpin forms just before terminator region, which causes the DNA to pause. Row then makes DNA and RNA to unwind and dissociate, releasing mRNA. Translation is the assembly of proteins based on mRNA coding. Three events must occur when initiating translation. A small subunit of the ribosomes, 30, is recruited to mRNA. The tRNA must be placed in the P site, and the large subunit of the ribosomes, 50, must be placed over the start codon. mRNA then binds to small subunits in the ribosomes. These subunits are bound to ribosomes by consensus sequences, called ribosome binding sites, also known as RBS. The RBS is upstream the AUG. The first tRNA carries formulated MET, which is attached to the first amino acid and coded by AUG. It must enter the P site. For initiation to begin in prokaryotics, there are three initiation factors. IF1, which keeps tRNA from binding to A site, IF2 provides energy, and IF3 temporarily stops large subunits from binding. Elongation begins when a second tRNA that is attached to an amino acid comes into the A site on the ribosomes. When putting together the polypeptide chain, termination consists of three stop codons, UAG, UAA, and UGA. They are recognized by release factors, which are enzymes that can mimic tRNA. During termination, the RNA strand and all subunits and proteins dissociate from each other. The newly made polypeptide chain begins to fold itself together. Lastly, amino acids are what make up proteins. These proteins can be folded in four ways. One way is primary structure, which is a long chain of amino acids. A secondary structure of protein will occur when hydrogen bonding occurs between these amino acids. These can be alpha helix or pleated sheets. Tertiary structures occur when the alpha helix and pleated sheets are attracted to each other. And quaternary structures is a protein with more than one amino acid.